Thank you for choosing my series, Seeing How We See, and the current section, Nerve Impulses. This video should help you understand neurons, nerve impulses, and how they work, and provide us with vision, motion, sensations, and everything that contributes to the experience of life that makes us us. Animals typically respond to stimuli using specialized cells called neurons. In this drawing, the stimuli is produced at A after this young man noticed a pretty girl walking toward him. The image of the girl was focused on the retina of each eye over millions of rods and cones that picked up the image. Each rod and cone produced a nerve impulse that represented the light falling on it for the small part of the image falling on that particular rod or cone. This nerve impulse is passed on to the first of a chain of neurons at B and then on from one to another neuron until it finally reaches the brain. In the brain, which is actually another large collection of neurons, this nerve impulse is combined with all of the other millions of neurons that allow him to see this girl in all her splendor. But the brain doesn't stop here. It sends several other nerve impulses back to the man's eyes, mouth, and the rest of his body to produce the typical reactions of a young man as he spots a pretty girl. Since neurons are involved in one way or the other in all of our daily activities, let's take a look at how they are actually doing their jobs and their makeup. Neurons transmit information throughout the body with changes in the electrical potential of the membrane surrounding the neurons. These changes are affected by movements of ions across the membrane into and out of the neurons. At rest, there is a larger concentration of sodium ions on the outside of the cell than the inside, and there is a larger concentration of potassium ions on the inside of the cell compared to the outside. The concentrations balance out so that the resting membrane potential is around minus 70 millivolts. The cell membrane is a phospholipid structure that does not allow easy passage of ions through it. Since the movement of ions in and out of the neuron is required to provide the passage of nerve impulses, several different ion-specific proteins are embedded throughout the membrane that provide ion pumps and channels that are used in coordinated arrangements to transfer ions in and out of the neuron as needed in order to generate and transfer nerve impulses through the neuron and to maintain the normal resting potential. On the left of the image, an active sodium-potassium pump, labeled A, uses the energy obtained from the conversion of ATP to ADP as it pumps 3 positive sodium ions at a time out of the cell, followed by 2 positive potassium ions into the cell, losing 1 net positive charge inside the cell as this is done, since 3 positive charges out minus the 2 back in result in 1 less positive charge inside, making the membrane potential slightly more negative during each cycle of the pump. The voltage-gated sodium channel, labeled B, opens when the membrane potential reaches a threshold value of minus 55 millivolts to allow sodium to move through the membrane. This channel also has a deactivation gate that blocks the passage of sodium ions after the voltage peak is reached, which prevents extra unwanted passage of sodium ions until current operation is completed. Another sodium channel, labeled C, is a ligand or chemically gated sodium ion. It plays a large part in the dendrite where nerve impulses from other neurons are received. A voltage-gated potassium channel is shown at D, which controls the flow of potassium ions through the membrane when membrane potentials reach their maximum values. At E, a voltage-gated calcium channel is shown that allows calcium in the axon terminal to flow when the membrane voltage reaches its peak positive value. The calcium causes a neurotransmitter chemical or ligand to flow to another neuron triggering its ligand-gated sodium channel at D to open and send a nerve impulse to the receiving neuron. At F and G, sodium and potassium leakage channels are shown. These are always open enough to allow sodium and potassium to leak across the membrane very slowly in response to the current voltage and concentration gradients that are existing across the membrane. They serve to set up and reset the membrane potential to its resting state and to shape the action potential when nerve impulses are being processed. The resting membrane potential of a neuron is established by the unequal distribution of sodium ions outside the cell and potassium ions inside the cell after adjustments occur that establish the equilibrium while the various protein channels are allowing ions to move through the membrane as needed. The membrane potential is stated as the voltage difference, with the outside of the neuron being considered the reference point of 0 millivolts. A typical value of resting membrane voltage is minus 70 millivolts. When an axon from neuron A passes a nerve impulse to the dendrite in neuron B, a neurotransmitter or ligand moves onto the membrane of cell B. This causes the ligand-gated sodium channel C to open and sodium ions rush into the cell. This causes the membrane voltage to rise towards a maximum positive value, typically plus 40 millivolts. This process is called depolarization. This jump in voltage is called an action potential that will move down the neuron to its axon terminal, where it will pass the nerve impulse on to the next neuron in the same manner as it was received from neuron A. The process of the action potential moving through the neuron will be discussed after a few basic requirements for this movement are discussed. As the dendrites of the neuron receive impulses from one or more neurons, the voltage changes drift through the neuron body into the axon hillock, where the incoming potentials from all of the neuron's dendrites are summed with the normal resting potential. If the resulting sum is less than the threshold of minus 55 millivolts, nothing happens. If the summation is greater than the threshold of minus 55 millivolts, an action potential is triggered, and through a series of ion exchanges through the membrane, an action potential is triggered that causes the membrane potential to rise to a maximum potential of around plus 40 millivolts. As the ions spread out along the axon to reduce the localized high concentration and the repulsion of the large number of positive charges, many leak out of the cell through the sodium and potassium leakage channels that are distributed along the membrane. The membrane potential at plus 40 volts millivolts at point A drops steadily toward the right as the concentration of ions and charges are reduced because of the spreading of the ions and the leakage out of the cell. As the charges spread out over the neuron, once they reach point B, the potential has dropped to plus 10 millivolts in this example. Slightly later, the moving charges reach point C and the voltage has dropped to minus 20 millivolts. Finally, at point D, the effects of the pulse of positive voltage at point A have diminished to 0, and the membrane potential is undisturbed and remains at the nominal membrane resting potential of minus 70 millivolts. Thank you for watching my first video on neurons and nerve impulses in the ShepNotes series. Watch for my next video to finish up our coverage of nerve impulses.